Hello everyone, welcome to Agriculture. Today we will be talking about something called restriction endonucleases, or more popularly known as restriction enzymes. Now if you are a biology student, you have probably heard the term several times in nuclear studies. There will be a full series of upcoming videos online, every video with 3 or 4 topics on restriction enzymes. So before we begin, make sure this video contains your topics of interest. A quick context for you. The topics of interest will continue to develop in the naming itself, the naming of restriction enzyme, the definition, and the background of discovery. What I mean to say is history and then evolutionary origin. Let's begin with the term restriction endonuclease. Well, not like this. Maybe like this: restriction, endo, and nuclease. What does endo mean to you? Endo means inside, and nuclease is an enzyme that cleaves the chains of nucleotide in nucleic acids into smaller units. What it really does is it takes a DNA, you can see a DNA molecule with 2 strands and GATTC and the complementary CTTAAG, and cuts it to be this one. Fair enough? So let's go back to this. Now, restriction endonuclease. So endo means inside, nuclease means this. So restriction endonuclease or restriction enzymes are something that cuts the DNA from inside. There are many molecules or many enzymes that cuts the DNA from outside, or say there is a strand like this. So it can cut on the ends of the strands, but it don't, because by that it will not be a endonuclease anymore. So it cuts from inside and makes this. Now the left is restriction. Restriction means to prohibit, and there is a little history out there. We will be talking about that. But before I really begin, let's compare our idea, what we have got, to the definition. A restriction enzyme or restriction endonuclease is an enzyme that cleaves DNA into fragments at or near specific recognition sites within the molecule, known as restriction sites. Fair enough? Now move to the history or the background. As I said before, it was early 1950s. There were 2 scientists. They were known Salvador Luria and Giuseppe Bertini. This is Italian, don't go with the spelling. What they found out is there's a tail between E. coli and fudge or lambda fudge. So there is a tail, or there is a relationship between E. coli and lambda fudge. The strains of E. coli have something to control, or some control on the growth of lambda fudge. What was seen was a E. coli strain, say E. coli C, when grown with fudge was growing perfectly. But another strain, say strain K, E. coli K, when grown with lambda fudge, the yield of lambda fudge dropped by 3 to 5 orders of magnitude. So by this, the scientists thought that the E. coli cells must have something in them, some molecule or something in them, what restricts the growth of lambda fudge. Now the term restriction endonuclease, if we concern, now the language restriction makes some sense, right? So in 1960s, it was shown in the work done by the laboratories of Werner Arber and Matthew Michelson, 2 scientists, popularly known as Arber and Michelson, that the restriction is caused by an enzymatic cleavage of the fudge DNA, and the enzyme involved was therefore termed as restriction enzyme. Well, later on in 1970, Hamilton Smith, Thomas Kelly, and Kent Wilcox, another three scientists, popularly known as by their surnames Smith, Kelly, and Wilcox, isolated and characterized the first type 2 restriction enzyme, HIND3 or HIND2. Now, we will be talking about the types and examples of restriction enzymes later on the upcoming videos. From the bacterium Haemophilus influenzae. Now, make sure you note down that, because in many exams, competitive exams like MCQ, in MCQs you will be asked. So make sure you remember that the first restriction or the first type 2 restriction enzyme that was isolated and characterized was HIND2 or HIND2. It was isolated from the bacterium Haemophilus influenzae and by Smith, Kelly, and Wilcox. Later, Daniel Nathans and Kathleen Dana showed that cleavage of simian virus 40, popularly known as SV40, DNA by restriction enzymes yields specific fragments that can be separated using polyacrylamide gel electrophoresis. Now, this crucial term polyacrylamide gel electrophoresis will be discussed on the upcoming videos. For now, you have just to know that this is somewhat a process by which the DNA can be mapped, thus showing that restriction enzymes can also be used for mapping DNA. This is also very important for competitive exams, and very important for the MCQs and all. And this fetched them the 1978 Nobel Prize. Whoa! Now, let's talk about the origin. By origin, I mean evolutionary origin. So, origin of restriction enzyme. If you search the origin of restriction enzyme in any book or any web page of restriction enzyme, you will get three terms or 3 points. The first is they are originated from a common ancestor. So, from a common ancestor, then spread via horizontal gene transfer, and then evolved as a selfish genetic material. But the common thing is that they never explain what is what. So you don't understand them, you have to mug up them. So we will be talking about this now. The first point, from a common ancestor. Now, what is an ancestor? Ancestor is some organism that stands before the organism in concern in the pathway of lineage. So ancestor is basically that organism or that molecule or anything from which our organism or our concerned organism came or evolved. Now, the second point, spread via horizontal gene transfer. What is horizontal gene transfer? Now, you have some idea about vertical gene transfer. Let me explain it to you. There were two parents, they had their own genes, they were crossed, and they made the filial generation or F1 generation or whatever, the next generation, their sons. So, the son has some genes from both the parents. These kind of gene transfers are known as vertical gene transfer, whereas horizontal gene transfer is transfer of gene or genetic information or genetic material between the organisms of the same generation. Say transduction, transformation, or anything like F pilus or something, they are all horizontal gene transfer. If you don't know anything about the transduction, transformation or something, don't worry, I'll teach you later on. And the third point is evolved as a selfish genetic element. Now, what is selfish? Selfish is something or someone who just thinks about himself and only himself. So these elements or these molecules, restriction enzymes, make more of the copies of themselves than required. So this more copy making phenomena has no role in evolutionary growth or evolutionary pathway, but they are made because the restriction enzymes wants to make sure that they are safe. So they make more copies of themselves, and which is literally a garbage. So scientists are finding, research work is going on to find out if this extra copy making phenomena has any lethal role on the organism. Well, this was the end of the video, and if you like the video, then you can like. If you don't, then don't, but do hit the subscribe button, because by that the channel runs literally. And make sure you hit the bell icon to never miss another update from every culture. Thank you.